Hi there, my name is Marissa Estrada. This is Lab 10 results video. It's Biology 225 of the Fall 2020 semester, and I forgot to write the date on this, but it is December 18th. Let me double check December 18th, 2020. I started this lab last night around 11:30-ish, 11:30 p.m., and right now it is 4:36 p.m. So, it's been over 12 hours. I checked it around 10:11, and there wasn't a lot of film on it. The results say to look for a white film, but there wasn't a lot. But now there's definitely growth. I'm not sure. Yeah, oh yeah, you could definitely see that. There is some growth. So this is the gentamicin. This is the novobiosin. I believe that's how you say it, and the penicillin. So if you can see, in my my hypothesis, I hypothesized that the penicillin would work the best. I was clearly wrong. So the gentamicin worked the best at inhibiting the colonies, followed by the novobiosin, and then last was the penicillin. Yeah, I was pretty inaccurate. Okay, so I forgot to mention, before I started, I sanitized my workstation with my 10% bleach solution and washed my hands and donned my PPE. Let's see. So I'm going to read through my lab, and so that way you know where I'm at within it. Wipe down your work area with bleach. Wash your hands thoroughly. Put on gloves, goggles, face mask, and apron. Gather the developed Kirby Bauer Diffusion plate and metric ruler. Take a photo of your plate. Let me try without the flash because it's just getting the condensation. There we go. Oh yeah. I wonder if you could see it in this. Yeah, so there is the bottom one down here is gentamicin, and then the novobiosin, and then last is the penicillin, which you could clearly see did not do very well in inhibiting the growth of the S. epidermidis. So I'll upload that to the HOO website once I'm completed with this video. Let's see. Without removing the lid to the plate, measure the zone of inhibition around each disc by placing the ruler over the center of each disc and measuring the diameter in millimeters of the zone with no bacterial growth. Okay, I'm going to write this down here. So penicillin. So that's about 1.1 centimeters, and then I'll convert that to millimeters. So next we'll do the novobiosin. Let me use my Sharpie actually because it's kind of hard to see. Penicillin was 1.1 centimeters, which I need to convert to millimeters. And then the novobiosin got stuck in my, there we go. Okay, so now for my novobiosin. It's about 2.5 centimeters, which I will convert to millimeters. And then the last is the gentamicin, which is 3.2. About 3.2 millimeters, or centimeters, excuse me. Okay, so I'll record those measurements to data table 1, and then I will be putting this into my bucket of bleach. And then I think that's it. If I could do anything different in this lab, let's see. I can't think of anything. I feel like the lab went pretty well. I also did the microscope. I'm pointing to the microscope. You can't see it. I don't know why I keep doing that. I did the microscope as well in this lab, which I was looking at the different structures of the penicillium, which was pretty interesting. I can show you my photo that I took of that the other night. Let's see here. Well, last night, it wasn't the other night. So this is the one that I ended up using. All the different, if I block my face, all the different structures. So here is the, what is that called? I'm trying to think what it's called. So, but you could see all the different structures. Like I could clearly point them out, and I was able to label them on the photo. So I ended up labeling them on my laptop and putting them into the photo. So if you look over here, so this is a string. Let me double check that. Yeah, so this right here, the string of conidium. I believe the plural is conidium, and then if you look very closely, the little dots are single conidia. And then let me grab my lab so I can give you the correct terms. I don't want to just be spouting off stuff. Okay, so, okay, I was incorrect. So if you look here, get my thing to focus on it again. Okay, so these branch-like filaments are the phyllide, which, oh my goodness, I'm sorry. Okay, there we go. So all these little branch-like filaments down here are the phyllides. Then, if you look closely, this is a very good example right here. This string of dots is considered the conidia. So I was incorrect. The conidia is a string of dots, but the single dots are the conidium. Okay, I think that's about it for this lab. So my intentions now are to soak this in bleach for 2 hours before dumping it in 100% bleach, and then I will be uploading my observations to my HOL website, and I will be sanitizing everything. Okay, that's it. Have a wonderful day.